This conference will now be recorded.
Okay, in the last class we discussed about the regulatory enzymes. In that we discussed about the different types of regulation process that are mainly involved in the regulation of enzyme in metabolic pathways. Okay, in that we discussed mainly about the allosteric regulation, and in that allosteric regulation mainly it is caused by the substrate affinity. That means substrate is involved in the regulation. Okay, and also ATP, CTP, these are involving in the regulation of the enzyme called as aspartate transcarbamylase. And next, here in this, the enzyme regulation that also mediated by covalent modification. In the covalent modification, different functional groups and that are attached to the enzyme. And here, some molecules are acting as the transfer of functional groups to the enzymes, and that causing the modification of the enzyme, and that causing the inhibition or increase in the activity of catalytic activity of the enzyme. That is called as a covalent modification. In that we discussed about the example of glycogen phosphorylase, and this glycogen phosphorylase that is mainly involved in the process of glycogenolysis, and here phosphorylation, dephosphorylation, and that are the different types of reactions that are involved in the regulation. And also ADP ribosylation, adenylation, and these are the covalent modifications and that are occurring in the process. Okay, and next here, that means here these are the regulatory enzymes, that means these are controlling the enzyme activity, and these all we discussed. Okay, covalent modification in that phosphorylation, main example is the glycogen phosphorylase, and ADP ribosylation, glycosylation, adenylation, and here, and why these covalent modifications are important for the metabolic pathways. All this we discussed. And next we are going to the here next type of regulation, and that is the control of enzyme availability. And that means here, if the enzyme efficiency, that means it is available, means the process occurs, the metabolic pathway occurs. Whereas if the enzyme and that undergoes degradation, means there is no activity, there is no metabolic pathway is occurring. And that means here the cellular physiology, that means here physiology means here different metabolic pathways are involving in the process, and these metabolic pathways and differentiation process, and these are requiring the enzyme at different steps. That means here at the starting or at the ending or different stages, this enzyme activity is required for the metabolic pathway to occur. Okay, and that means here the process of enzyme turnover. That means here turnover means, what is the turnover, ma? Here, what is turnover? Already we discussed about this topic. Turnover means, yeah, turnover means, yeah, number of substrate that are converted into a product in a particular time. And that means here that enzyme turnover is necessary for adaptation of the cell to undergo the changes in the environment. And that means here after the completion of this turnover, here that means the substrate is converted into the product at a particular time, in a particular time, means here that means it is adapting to certain changes, and that may be environmental changes or any changes. After that, we have to remove that particular enzyme from the process. Then only the metabolic pathway is efficient. Okay, and here after the process we have to remove that particular enzyme. That means here this enzyme undergoing several modifications during the process, and that must be removed. Okay, and here, and that occurs in different steps, and also here the enzyme regulation is also occurring at gene level. Gene level means, and that is at the transcription level or or it is at the translation level. The regulation of enzyme also occurs at the gene level, that is a transcription level, or at the translation level. And in this, mainly here, the peptide bond hydrolysis in enzymes. In enzymes, here, the process of peptide bond hydrolysis, that means it is occurring in translation process. And in translation process, what is happening? Some of the steps requiring energy and some of the steps that are not requiring energy, and that is liberating the energy in the form of ATP. And here, and that process of peptide bond hydrolysis in the enzyme, these are not liberating the energy. Whereas peptide bond formation, and that is utilizing the ATP. That means here, depending upon the environmental condition, here the enzymes, these are changing in their activity. These are changing in their activity. That is, this activity has to be reduced for the controlling of enzyme activity. Okay, and that means here that enzyme must be degraded, and different types of degradation process are there for enzyme degradation. And what are the process? And here, actually, this process, the protein degradation, means that mainly protein means here, and that may be mainly at the translation level where the protein synthesis occur, translation level. And that means here, and that protein degradation where it occurs means after the synthesis of proteins. Here these are transported to endoplasmic reticulum, Golgi complex, and different cellular organelles are involved in the this protein formation. Okay, and here the degradation, and that is mainly occurring in lysosomes. Degradation process mainly occurs in the lysosome. And here, and that requires a proteasome complex. That requires a proteasome complex. That means here the protein degradation occur both lysosomally and proteasomally. That means here the proteasomic complex for the degradation of protein is necessary in which organelles? Lysosomes. In which organelles? Lysosome. And that process may be energy dependent or it may be independent. Energy dependent process or it may be energy independent process. Or it can be selective process or non-selective degradation. That means if particular proteins has to be degraded. That means here enzymes means here we are discussing about the proteins. What are proteins means that are of enzymes. Okay, and here these can be, that means the selective degradation process may be present or non-selective degradation process may be present. Okay, and here, and these enzymes, these are mainly degraded by the lysosomal degradation process. And here in the enzymes, different types of enzymes are present, and that means depending upon the time period. Here these are classified into housekeeping enzymes and short-lived enzymes. What is meant by housekeeping enzymes? What is meant by housekeeping? Anyone? Yeah, always express. Good, Masha. Here, housekeeping. Housekeeping means this is a service, housekeeping service. And that means housekeeping means it always occurs. And that means these are the enzymes, these are always expressed. And these ought to be continuously synthesized and continuously degraded by special type of degradation process. And that means here many housekeeping enzymes, and that ought to be degraded by lysosomal degradation process. Lysosomal degradation process. Whereas the another type of enzymes are present, that means these are called as the short-lived enzymes that has a short lifespan, and these are degraded by extra lysosomal mechanism and are present in different organelles. And these are present in different organelles. That means here proteasome complex, and that is mainly involved in that type of enzyme degradation. In that type of enzyme degradation process. Okay, and here in that means here this is a proteasome degradation process. That means here this is called as the enzyme degradation process, and this is called as irreversible covalent modification. It is called as irreversible covalent modification. And what is here also modification of the enzyme occurring, but it is irreversible covalent modification. Okay, and that means here the enzyme, sometimes the enzyme must be synthesized in the form of inactive state, in the form of inactive stage, and then it is activated by several cleavage processes. And that means here these enzymes are nothing but proteins that contains a different peptide bonds, and these are to be degraded, and that means the peptide bonds are broken, and that causing the activation of these enzymes, activation of these enzymes, and that inactive enzymes and that inactive precursors, and these are called as zymogens. The inactive precursors are called as zymogens or proenzymes. Proenzymes. And here, what is happening? These are to be converted to be active state. That means inactive state to active state must be converted for the activation of the enzyme. And that activation process occurs by the degradation process, and that degradation occurs at the peptide bonds. And that means here the proteolytic cleavage is the main process that is involved in the activation of the enzymes, and that is included in irreversible covalent modification. Irreversible covalent modification. Here, the peptide bonds are cleaving for the activation of the enzyme, and where this cleavage occurs, and that cleavage, it is generally occurring at the surface loops only, at the surface loops only. And here in proteins different conformations are present: primary, secondary, tertiary, quaternary. Different conformations are present, and that means different elements are responsible for the conformation of proteins. And here the proteolytic cleavage, it is specially it is occurring at the surface loops only. It is not interfering with the structural conformation, that is secondary structural, tertiary structures. Okay, and here, and that proteolytic site, it is mainly of amino terminal site. Amino terminal, and that is relating to the active site of the protein. That is relating to the active site of the protein. And why means here actually the polypeptide synthesis, the protein synthesis occurs from amino terminal to carboxy terminal direction. And that means here mainly the proteolytic site is the amino terminal related to the active site of the protein. Okay, and here the polypeptide is synthesized in amino to carboxy direction means here, and that means here the foldings that are responsible, the general surface loops, and that are responsible for the process, and these are degraded by the proteolytic process. These are degraded by the proteolytic process. Okay, understood ma here? And that is called as a irreversible covalent modification, and that is also called as the limited proteolysis process. Limited proteolysis process, and it is coming under the protein degradation process ma. It is coming under protein degradation process. And in this mainly, for these main examples are digestive enzymes. Digestive enzymes. And normally these digestive enzymes, these are present in inactive state. These are present in inactive states, and the enzymes that are responsible for the activation, and these are to be present for the activation of these. Okay, and here the digestive enzymes are main examples of the enzyme activity that is occurring by specific special cleavage process. Okay, and here these are synthesized in the form of inactive precursors. Inactive precursor, and these are to be activated. These are to be activated. And how these are activated means some of the degradation process is occurring for the activation of these enzymes. And where these enzymes are synthesized means here the site of synthesis is stomach, mainly in the stomach pepsinosin, and this is synthesized in the form of inactive state, and the active form of this is the pepsin. Whereas in pancreas mainly these three types of enzymes are present in the form of inactive state, and these are to be converted into the active state. That is chymotrypsinogen, trypsinogen, carboxypeptidase, and these are to be converted into the active enzyme chymotrypsin, trypsin, carboxypeptidase. And also in pancreas, proelastase, and this is synthesized in the form of inactive form, and this is to be converted into the active enzyme that is called as the elastase enzyme. And these are the different types of digestive enzyme that are responsible for the uh, activation of enzyme activity by specific cleavage process. Specific cleavage process. How this activation occurs, I think you know about this. And in the digestion process, these all enzymes are coming. Okay, these are called as the digestive enzymes here. These are to be converted into the active form by different enzymes. Here, trypsinogen is converted into the trypsin active form by the enterokinase enzyme. And this trypsin, again, it is involved in the activation of trypsinogen, chymotrypsinogen, proelastase, and procarboxypeptidase to trypsin, chymotrypsin, elastase, and carboxypeptidase. Just a minute, ma'am.
This conference will now be recorded.
Yeah. Okay. Up to this we completed, that is enzyme degradation. And next coming to the digestive enzymes. Here in the enzyme degradation, the main example for enzyme degradation process is digestive enzymes. And in digestive enzymes, and the activity of the enzyme, and that occurs by a specific cleavage process, and the cleavage occurs at the peptide bonds. The cleavage occurs at the peptide bonds, and that causing the inactive zymogens or proenzymes to the normal active enzymes. And what are the different these type of enzymes here means, and these are the different types of zymogens, and that are of mainly pepsinogen, chymotrypsinogen, trypsinogen, okay, and procarboxypeptidase, proelastase. And these are the different types of the enzymes, and these are synthesized in the form of inactive precursors. Inactive precursors. And these are to be activated for the continuing of the process, for the continuing of the metabolic process, that is the digestion process. And here, and these are to be converted in the form of active, and that occurs at different sites, and mainly the stomach, pancreas. These are involved in the digestion process, and that causes the activation of these pepsinogen, chymotrypsinogen, trypsinogen, procarboxypeptidase, and proelastase to the active enzymes. How this process is occurring? That is by degradation process. That is by degradation process. And how this activation occurs? That is activation of pancreatic protein. And that means here trypsinogen, and trypsinogen is the inactive zymogen, and that is activated by the enzyme enterokinase, and that is causing the conversion of trypsinogen into the active trypsin. And this active trypsin, and it is again involving in the conversion of inactive trypsinogen, chymotrypsinogen, pro-elastase and pro-carboxypeptidase to trypsin, chymotrypsin, elastase and carboxypeptidase. Okay, and here, and enteropeptidases. What are these enzymes? Enteropeptidases cause the conversion of inactive trypsinogen to the trypsin, and that trypsin that is causing the conversion of these inactive chymotrypsinogen to chymotrypsin, pro-elastase to elastase, pro-carboxypeptidase to carboxypeptidase, pro-lipase to lipase. Okay, and that means here due to the degradation of these, due to the degradation of the inactive enzymes, here these active enzymes are produced. Active enzymes are produced. And here this is the inactive trypsinogen, sorry, inactive chymotrypsinogen here, and that contains the amino acids in the range of 1 to 245. 1 to 245. And this here, this is the site that causes the cleavage. This is the cleavage site for the activation of this chymotrypsinogen, and that is caused by the trypsin. This is caused by the trypsin, and again this is activated by chymotrypsin, by chymotrypsin, and that is producing the alpha chymotrypsin, and this is the main active enzyme. This is the main active enzyme. Here two cleavage sites we are observing here, two cleavage sites. These are the two cleavage sites, and that means here the proteolytic degradation is mainly responsible for the enzyme activity in different metabolic pathways. It is also a type of regulation. Okay, understood ma here? Allosteric regulation is the one of the method. Next one is the covalent modification. In the covalent modification, reversible covalent modification is there and irreversible covalent modification is there. In reversible covalent modification, that is changing of the functional groups. Whereas in irreversible covalent modification means that is undergoing the proteolytic degradation process, that is undergoing by proteolytic degradation process. And another process is involved in the inhibition or activation of the enzyme activity, and that is called as the feedback regulation or feedback inhibition process. And what are these processes? Feedback regulation is there, feedback inhibition is there. And what is the difference between these? Anyone? Feedback regulation and feedback inhibition. Here, feedback means here, feedback, that means after the completion of your session, you have to fill the feedback, and that means here, the efficiency of that lecture, and that is given in the form of feedback form. Okay, and that means here after the completion, after the production of the product, after the production of the product, and that means the sufficient amount of product is produced or not, and that is detected by using the feedback, that is detected by feedback. And here feedback regulation is there and feedback inhibition is there. What is the difference in between these? Regulation and inhibition. Anyone? What is the difference between regulation and inhibition? Anyone? Yeah. Regulation means, yeah, good. Regulation means to maintain and inhibition is to stop. Okay, good. Inhibition means when enough of product is produced, the enzyme is inactivated. Okay, good. Manasa, regulate, initiates a process. Okay. Okay, I will tell ma here what is feedback regulation. What is meant by feedback inhibition means here feedback regulation, regulated. That means we are using the gas stove and that is controlled by regulator. Yeah, here that means that is controlling the gas. Here also after the production of the product and that product that is causing the regulation of the enzyme, that is causing the regulation of the enzyme. That means here in the feedback regulation, mainly the end product that is produced by the conversion of the substrate by the enzyme and that causing the decrease in the rate of enzyme synthesis at the level of gene expression. At the level of gene expression. That means it is causing the decrease in enzyme synthesis. That is decreasing. That means here that is regulating the enzyme synthesis. Okay, and that means it is not affecting the enzyme activity, just only regulating the enzyme activity. And that means it is not affecting the enzyme activity. That means it is decreasing the enzyme quantity through the action on the gene that encodes the enzyme. And that means at the level of gene expression means here the gene that is responsible for the activation of the enzyme that is regulated by the feedback regulation, by the feedback regulation process. And for this main example is the inhibition of HMG-CoA reductase by dietary cholesterol. Dietary cholesterol. And next coming to the feedback inhibition. What is meant by feedback inhibition? In the feedback inhibition, what is happening? The end product that is directly inhibiting the enzyme and that is present in the biosynthetic pathway, that is the early enzyme that is affecting the early enzyme in the biosynthetic pathway. Early enzyme is here the first enzyme that is involved in the metabolic pathway and that is inhibited by end product, that is inhibited by end product, and that means it does not affecting the enzyme quantity but it is decreasing the enzyme quantity by the end product, and it is a direct and a rapid process that occurs within a second. It occurs within a second, and that is the difference between feedback regulation and feedback inhibition process. Okay, and also feedback repression is there. Anyone? Feedback repression. Yeah, Deepshika, I will repeat again. Okay, don't worry, ma. Anyone? Feedback repression. Yeah. Here feedback regulation, that means regulation, and that is means regulating the activity of the enzyme. That is called as the feedback regulation. Understood ma here? And it is regulating the activity of the enzyme at which level means at the level of gene expression. The gene that is responsible for the synthesis of the enzyme and that is regulated by feedback regulation, by feedback regulation. Whereas in feedback inhibition, that means it is completely inhibiting the enzyme, inhibiting the enzyme in that particular pathway, in that particular pathway, and that is called as the feedback inhibition. Understood me? What is the difference here? Regulation means regulated. That means up to that. That means for certain time. That is the enzyme activity is regulated. Whereas inhibition means and the loss of complete enzyme activity occurs during the inhibition process. Deepshika, is it okay, ma'am? Yeah. Okay. Next, feedback repression is there. What is meant by feedback repression? Feedback repression. I think you know. Lacoperon is completed in your syllabus. And what is here in lacoperon? Repressor proteins are there. And here, and that repressor proteins that are mainly involving in the regulation of the transcription. Yeah, and that means here also enzymes. These are acting as a repressor proteins. These are acting as a repressor proteins for the regulation, and that means that is called as the feedback repression means here repressor proteins. What are the repressor proteins here? Enzymes are acting as that means here enzymes, and that are acting as the repressors in the metabolic pathways, and that is inhibiting that type of particular reaction, and that is called as the feedback repression. Once again, I am repeating here. What is meant by feedback repression means actually in the feedback system, what is happening, and the enzyme activity is inhibited and is also regulated. These two processes are normally occurring. Whereas in repression, the end product and that is produced in metabolic pathways, and that causing the conversion of the enzyme as a repressor and causing the regulation and that is called as the feedback repression. And these are the different types of process that are involving in the regulation at the transcription level and at the translation level. Okay, understood here? Actually, 4 steps are there for the regulation of the enzyme activity. That are of mainly allosteric enzyme regulation. And second one is the covalent modification. And the third one is the at the gene level, that is the transcription level and at the translation level. And in the transcription level and in the translation level means here this feedback regulation and feedback inhibition process, and these are coming at that stage at the gene level. These are regulating or inhibiting the enzyme activity. These are inhibiting the enzyme activity, and that are called as the feedback regulation and feedback inhibition process. Understood, ma? And the next process is the isoenzymes. And what are iso? Iso means here similar ones. And that means here enzyme, and that is occurring in the form of multiple forms, and that is catalyzing the same chemical reaction but showing different properties but showing different properties, and that are called as the isoenzymes. What are isoenzymes here? Isoenzymes or multiple forms of the same enzyme, and that is catalyzing the same reaction, but their properties are different. Their properties are different. And what are the properties here that are different? That means here that are differing in physical properties and chemical properties. And what are the physical and chemical properties? Physical means, and that is mainly kinetic property. Whereas in chemical means, and that means the chemical composition of these enzymes, and that is depending upon that here, and these properties are differing. And what are the properties means here? Mainly electrophoretic mobility, amino acid sequence, amino acid composition. And these are the main properties, and that are differing. Isoenzymes are the multiple forms of the enzyme, and that are differing in physical and chemical property but are catalyzing the same chemical reaction. Understood, ma? And that are isoenzymes. And here, and what are the examples for these? That means the properties are different. That means different enzymes are responsible that are showing different types of properties. And what are the examples for these? Here, electrophoretic mobility. And electrophoretic mobility, for this main example is the isoenzymes of lactate dehydrogenase. Lactate dehydrogenase, and that shows different electrophoretic mobility, and based on that these are classified into mainly three classes: LDH1, LDH2, LDH3. Okay, and the catalytic reaction is same but showing different in electrophoretic mobility property. Okay, and next, depending upon the heat stability, alkaline phosphatases, and these are classified into heat labile ones and heat stable ones. Heat labile ones and heat stable ones, depending upon the property of heat stability. And next, coming to the inhibitor, depending upon the inhibitor, here this acid phosphatases, and these are classified. An inhibitor means that is inhibiting just only one isoenzyme, and that means based on the inhibitors, here different types of inhibitors are acting on different forms of acid phosphatase enzyme, and based on that here these are different. And also based on the Km, that means here this is a kinetic property, and based on that here glucokinase and hexokinase are there, and this mainly depends upon the affinity, low affinity, high affinity, depending upon the substrate affinity. These are classified into glucokinase and hexokinase. And next, based on the cofactors, also here, these are classified into mitochondrial isocitrate dehydrogenase and also cytosolic isocitrate dehydrogenase. And these are requiring cofactors, and these cofactors are differing. That means based on the cofactors, also here these enzymes are classified. That means these are catalyzing the same reaction. That means here dehydrogenase reaction occurring, but these are requiring the cofactors differ. Okay, NAD plus is the one of the cofactor that is required for the activity of isocitrate dehydrogenase that is present in mitochondria, whereas in cytosol that is requiring NADP plus. Okay, and also depending upon the tissue localization, and that means these isoenzymes that are present in different organs, and that are present in heart and that are present in muscles, and depending upon that also these are classified. And also antibodies that contains different types of antibody. For this main example is the creatine kinase, that means these are bound by specific antibodies only. These are bound by specific antibodies, and these are the different types of isozymes. Okay, multiple forms of the enzymes. And in this, first coming to the first example, that is a lactate dehydrogenase, and this is the enzyme classification. That means here depending upon the nomenclature process, and it is given in the form of enzyme classification and 1.1.1.27. Okay, and what is the function of this enzyme? Dehydrogenase means here, and that is requiring the cofactor NAD plus, and that is acting as the oxidoreductase. That is acting as the oxidoreductase. And here, and this lactate dehydrogenase, here what is the property that is classified for the classification of lactate dehydrogenase? It is mainly based on the tissue localization. Lactate dehydrogenase isozymes are classified mainly based on the tissue localization, that is, and that are present in different organs, different tissues. Okay, and here, what is the molecular weight here? And for this, the molecular weight is 134 kilodaltons, and it is occurring as a tetrameric composition. And here, and this is occurring in muscle and heart. And in muscle, that means in the muscle that is occurring means the enzyme that is present at the chromosome level. Chromosome level. Okay, whereas in heart means that is presenting at the chromosome 12 position. 12 position. And also here in the muscles it is occurring in the form of basic form, whereas in heart it is occurring in the form of acidic form. Hence this is called as NAD plus oxidoreductase. NAD plus oxidoreductase. Okay, and these are the isoenzymes for the lactate dehydrogenase. Okay, and this is the LDH1 and this is the main heat resistant one. Here, this is the lactate dehydrogenase 1, heat resistant one. Here it is occurring as the tetramer. It is occurring as the tetramer. Okay, and that is mainly present in heart. Hence it is called as H4. H4 means here that is occurring in heart in tetrameric form. And depending upon the that means electrophoretic migration, and this is the fastest moving in electrophoresis, and that is presenting myocardium, RBC and kidney, and it is elevated in myocardial infarction. And here this is acting as a marker for the diagnosis of diseases. At the diagnosis of diseases, and these isoenzymes, these are used as the markers. These are used as markers. Okay, and what is myocardial infarction? Anyone? Infarction, myocardial infarction. Yeah, good. Heart attacks, heart attacks. Good, ma. And here, and that means this acting as a marker when this concentration is elevated means it is a marker for the diagnosis of myocardial infarction. And also for the kidney patients, if you observe creatine levels are increased means, and that is prone to kidney diseases, and that is acting as a marker. And also that creatine levels, if more levels are present means, that is acting as a marker for heart attacks, for myocardial infarction. That means here these isoenzymes, these are acting as a markers for the disease diagnosis. Okay, and here LDH2, another isozyme, and that is a heat resistant one, and that is occurring in the form of composition of H3M1. Okay, and that is present in myocardium, RBC, kidney, and it is mainly elevated in kidney diseases and megaloblastic anemia. Megaloblastic anemia. And next, coming to the LDH3, it is occurring in the form of H2M2, and these levels that are mainly present in brain. Brain means that is causing the leukemia and malignancy. And next, coming to the LDH4, it is occurring in the form of H1M3, and that is mainly present in lungs and spleen, and that is causing the pulmonary infection. Okay, and next LDH5, and it is a heat level one, and it is mainly inhibited by urea, and it is occurring in the form of muscle M4, and it is the slowest moving one, and mainly present in skeletal muscles and livers. Okay, and that means it is a marker for the skeletal diseases and liver diseases. Okay, and this is all about the lactate dehydrogenase. And next, coming to the creatine phosphokinase, and this is a this is occurring in the form of adenosine triphosphate creatine N phosphotransferase. Okay, and the enzyme commission that is giving enzyme classification is 2.7.3.2, and it is occurring in the form of dimeric enzyme. And their international units, means the concentration is in the range. This concentration to be in the range of 4 to 60 international units per liter. Okay, and this creatine kinase and this enzyme, and this is mainly present in serum, prostate, lungs, uterus, brain, intestinal tract, urinary bladder, heart, skeletal muscles. Okay, and this is the concentration, that means it is occurring in many organelles, and this enzyme activity is needed for many organelles. And here, and this is classified into this creatine phosphokinase it is classified mainly into three types, and that are mainly occurring in cytosol. It is mainly occurring in cytosol. Okay, and here depending upon the tissue of origin, it is also classified. And here that means here this is the origin that is occurring from brain. That means it is occurring as the dimeric enzyme. Here it is occurring as the dimeric enzyme. That means here BB means that is mainly occurring in brain, and that is mainly present in brain, prostate, gastrointestinal tract, lung, bladder, uterus and placenta. And this concentration is elevated means that is giving the diseases called as the central nervous system diseases. Okay, and next CK2, that is a creatine phosphokinase 2, and normally this is a percentage, 2 percentage of all enzymes, and that is to be present in cytosol. And this is a marker for the myocardial diseases and heart diseases, and that causing the acute myocardial infarction. Acute myocardial infarction. And next is CK3, and that is a slow moving ones, and that is mainly present in muscles. Mainly present in muscles. That means here skeletal muscles and myocardial muscles. And these are mainly responsible for this activity of enzymes. Okay, and that is the creatine phosphokinase enzyme. And next coming to the alkaline phosphatase. Alkaline phosphatase, and it is occurring as orthophosphoric monoester phosphohydrolysis enzyme, and the enzyme commission that is giving in the form of 3.1.3.1. Okay, it is mainly present in small intestine, proximal convoluted tubule, bone, liver and placenta, and that causing the hydrolysis of naturally occurring and synthetic substrates. Okay, and this is a alkaline phosphatase, and this is a marker for mainly bone diseases. This is a marker for mainly bone diseases. Prostate cancer and bone diseases. Okay, okay. These are the different types the isoenzymes of alkaline phosphatase, and depending upon the heat level and stability. That means here these are occurring in the form of heat level nature and heat stable ones. And depending upon that, these are classified into alpha 1, alpha 2, in that beta, gamma, and leukocyte alkaline phosphatases. Okay, and these are the different types of alkaline phosphatases. And here, if alpha 1 or ALP that is elevated means that is present in which cells here? Epithelial cells of biliary canaliculi. Okay, any easy way means here, first of all you have to remember and what is the main disease that is occurring by these diseases, sorry, by these enzymes. For example, lactate dehydrogenase, you have to remember how means lactate dehydrogenase means that is occurring in tetrameric form, mainly in which organelles, and that is you have to focus on the organelles, and that causing the specific diseases, and that causing the marker for the specific diseases, and in that way you have to remember now, Asha. Okay, and next coming to the creatine phosphokinase. Creatine phosphokinase is elevated means, and that is causing mainly myocardial infarction means that is affecting heart. That means here first of all you have to remember the origin of this enzyme. That means here which organelles are responsible for the activity of this enzyme. Okay, in that way you have to remember, and then it is easy for that for you. Okay, and next alkaline phosphatase, it is mainly present in hepatic cells. It is mainly present in hepatic and intestinal cells. Hepatic and intestinal cells. Okay, and that mainly causing the diseases hepatitis and bone diseases. That is mainly causing the hepatitis and bone diseases. And in that way, hepatic cells means what are the cells that are responsible for their liver function, and that means here these hepatic cells means that contain the epithelial cells of biliary canaliculi, and some types of cells are presenting that liver cells means you can remember. Okay, and that means alpha 1 ALP, that is epithelial cells of biliary canal nuclei. Next, heat labile ALP, and that is mainly present in hepatic cells. And next means heat stable ones. Heat stable means it is not destroyed at high temperatures. It is not destroyed by high temperature and also inhibited by, but inhibited by some enzymes or some precursors. And that means here this is inhibited by phenylalanine. Phenylalanine. And next here, pre-beta ALP. And you have to remember this also, ma, and that is mainly present in bone. And that is the marker for the bone diseases. It is the marker for the bone diseases. Beta, pre-beta ALP is the marker for the bone diseases. And next, gamma ALP, that is present in intestinal cells and that is inhibited by phenylalanine. And next, coming to the leukocyte alkaline phosphatase, and that is mainly decreased in CML and it is increased in lymphoma. Okay, and that means mainly it is involved in the leukoma cells. Okay, and also atypical isoenzymes. Atypical isoenzymes, in that mainly two enzymes are present and that are Regan isoenzyme and NACU isoenzyme. And this is not necessary for human atypical enzymes. Okay, you have to remember up to this only. Alkaline phosphatases. Okay, these are the variants of these isoenzymes. These are the variants. These are the variants. Okay, and that are the alkaline phosphatases. And next coming to the acid phosphatases, and this acid phosphatases, it is mainly causing the disease called as the prostate cancer, and this is a marker for prostate cancer. That means here the main organelle that is responsible for this enzyme activity is the prostate, and that is causing the disease called as, and that means elevated levels of this enzyme means that is causing the disease called as the prostate cancer, and also present in bone, spleen, platelets and liver and kidney. Liver and kidney, and it is occurring in the form of acid phosphatases, and that causing the hydrolysis of phosphoric acid esters at pH 5 to 6 in lysosome. That means this is extra lysosomal enzyme. This is the extra lysosomal enzyme, and the enzyme commission that is given in the form of 3.1.3.2 as in. Okay, and that is acid phosphatases. Okay, and this is mainly used for the prostate cancer detection. Okay, and also here tartarate resistant acid phosphatases, and that causing the increase in Paget's disease and also bone cancer disease. Bone cancer disease. That means here this is mainly responsible for bone diseases and cancerous. In the cancerous, mainly prostate cancer. Prostate cancer. And that is causing the cell tumor of bone. Cell tumor of bone in normal growing children. Okay, and also Gaucher's disease. Gaucher's disease, and this Gaucher's disease, it is also coming in metabolic pathways, and that is in high concentration. It is occurring in high concentration in semen. Okay, and this is the clinical significance of acid phosphatase. Okay, and here what are the tumor markers? The main tumor markers. Okay, I'm just giving the brief on tumor markers, and tumor markers mainly aldolose. Aldolose is a marker for the liver diseases, and whereas alkaline phosphatase, it is a marker for bone, liver, leukemia, and sarcoma tumors. Okay, next placental ALP is increased means, and that is causing the ovarian cancers, lung cancers, and Hodgkin's cancer. Okay, and next amylase is increased means that is causing the pancreatic cancers, whereas in creatine phosphokinase, and that is increased means that is causing the prostate, lung, breast, colon and ovarian cancers. And next GGT means that is glutathione once more, and here that is acting as a liver cancer. Liver cancer. Okay, next lactate dehydrogenase, and that is elevated means that is causing liver, lymphoma and leukemia type of cancers. And next, neuron specific enolases. These are the markers of neuroendocrine origin. Neuroendocrine origin. And what is the function of these isoenzymes? That means here these are acting as a markers for the diagnosis of different diseases. And that means here these enzyme activity, why we are studying these isoenzymes in regulated part means here these isoenzymes, these are occurring in different forms, and their activity is mainly used for the studying of diseases, and that means the regulation of the enzymes, and that is controlled. The enzyme activity is controlled means there is no disease, whereas the enzyme activity is disturbed, that is causing, that is prone to the diseases. And that means here the enzymes, isoenzymes are also coming in regulatory enzymes, and these are the different forms of the enzyme, and that causing the enzyme activity. And when the activity of these enzymes is increased means, and that are prone to different diseases, and that are acting as a markers. Okay, and these are the different types of regulation processes. Allosteric regulation is the first one, ma, and the second one is the covalent modification. In the covalent modification, irreversible covalent modification, that is causing at the gene level. Okay, and that is the third one. And next, isozymes and feedback regulation, feedback inhibition. These are the different types of regulation process. Okay, understood, ma? All of you, is it clear? Yeah. Okay, Asha, Manasa, Dipshika, Mavunya, Vidushi. Okay, ma. And this is all about the regulatory enzymes, and that is mainly involving in the covalent. That means that is mainly mediated by different regulation process. Okay, here mainly what are these here? Mainly allosteric regulation, and next covalent modification, and next by regulated expression of gene or by synthesis of enzyme, and by regulated destruction of enzyme, and next it is also considering with the isoenzyme part for the regulation. Okay. Okay, ma. And regarding this, for these questions, we will discuss in the next class, ma. Okay.
Okay, thank you, ma'am. So, students, if you have any doubt regarding today's talks, please comment in the comment box. Okay. Okay, thank you. So, if you understood today's topic, please comment yes in the comment box. Just let us know. Okay,
After a long
thank
time,
you.
Prema Kumari is responding.
Yes, ma'am. I think she is sleepy at that class. Okay. Okay, thank you, everyone. So, tomorrow we'll meet. So now you can leave the meeting. And thank you for your response.